And so for today, I'll be talking about cotton, but really you can pretty much insert your favorite crop here in terms of the technology and the applications. And so this is where cotton is produced in the U.S. We're in Texas, which you probably already know is the largest cotton-producing state. So this is always good to be in Texas to give this talk. But in terms of where cotton is produced, it's produced along the southern part of the U.S. This is affectionately known as the Cotton Belt. And it has the unfortunate realization that this is where a lot of heat and drought stress occurs in the U.S. And so in any cotton-growing region, you're typically going to experience these adverse environmental conditions. And so 2011 here in the state of Texas, you guys had one of the worst droughts ever. $2.2 billion economic loss in cotton production alone. You guys lost $5.5 billion in cattle. And so additionally, 55% of the acres planted that year were just abandoned in the field. No attempt to even, save the crop at all. And unfortunately, global data supports the same pattern in other cotton-producing regions, whether it's China or India, that this is a continued problem that will be facing cotton breeders as well as cotton production, primarily, going forward in the future. And so the challenge is, how do we develop heat and drought stress tolerant varieties? Because unlike our, I can guarantee every one of you in the room is wearing a piece of cotton. Whereas, the same thing can't be said for what you had for breakfast, whether it was one of the major cereal crops. And although, cotton is not food, and I'm not going to stand up here and tout the benefits of fighting, world nudity, the fact is there is no economically sustainable solution for this fiber crop. And why, this is why, there's so much emphasis on how do we sustainably produce this fiber. And so really, heat and drought stress tolerant varieties is plant breeding. The drought phenotype is a whole plant response. And it's a phenotype that really relies on the environment in which it's expressed. And because of this, I would argue, and I think the literature really bears this out, that there really is no single gene solution. And, primarily we look at, there's so many components. I mean, in terms of photoprotection, we have the actual leaf architecture and the composition in terms of the pigments that make up the leaves. At the other aspect, there's transpirational efficiency, how water is being pulled through the plant, and the trade-offs that the plant must make in terms of losing water to fix carbon, and how vapor pressure deficit really drives that, demand. There's also water uptake, how the roots are exploring the ground to capture water, how it's being driven up into the plant itself, and during times of drought, how that root architecture explores deeper other areas to acquire that water that's needed. And finally, there's always the basic principle of partitioning. In terms of bulb production, leaf production, stems, roots. And so because of that, plant improvement really has to occur at the whole plant level. And to do that, we really need to understand the underlying biology as well as the genetics of stress response. And although primarily I focus on transpirational efficiency and water uptake kind of in my research, it's really all these components together that, give a plant its drought resiliency. And so that's kind of the challenge we're facing now, is how to integrate physiology and genetics to really meet the demands of the plant as well as, future crop plant production areas. And so kind of illustrate this at another level is that when we think about genotypic information, it's really bit information. It occurs in two dimensions. It's A's, T's, C's, and G's. And, for the past 20 years, we've gotten really good at, finding these, cataloging them, storing the data. But really, it is just a two-dimensional space. And it's through the interaction with the environment that we add this complexity where you can see finally in this final figure, we get these, phenotypic optimums, just like any selection paradigm, where there's an optimal phenotype for different environments. And it's only, through this interaction of the genes with the environment that these arise. And so the challenge is how do we, find these optimal phenotypes for the different environmental challenges. Questions that guided this research, first from an engineering standpoint is it possible to actually collect meaningful, physiologically relevant data at field scale under field conditions? In terms of more of the applied breeding, is there exploitable variation for stress adaptive traits in cotton? We all know that you have to have variation. That variation must be heritable. So does that exist? more just a basic genetics question is there a temporal basis of QTL expression patterns in cotton? You know, this idea of when genes are turned on in relation to the environment, as well as actual plant development. And then finally, very applied, you know, can we use physiological trait data to actually predict agronomic performance? So obviously, I would make the argument that high-throughput phenotyping is an essential tool, and that it gives us the ability to evaluate large populations under field conditions. And this is imperative for drought research because there's such a dynamic interaction between the environment and the plant that, you know, soil crusting, hardening, you can't really replicate those exact same conditions in a greenhouse environment. Also, you know, typically the results you see in a greenhouse don't generally translate to the field environment as well. So that's why having this field component is so essential. Additionally, we're able to measure the trait throughout development. Instead of just taking these static time point measurements and only seeing really what's happening at that one point, by longitudinally assessing the phenotype, we actually get more of a video of what's going on, how the trait's developing in relation to phenology as well as the environment. From a research standpoint, we can work with larger genetic populations to increase power for genetic mapping, GWAS, genomic selection as well. And I would also argue that there's a lower cost and minimized subjectivity. You know, as field breeders, you're walking through, you're scoring plots as fast as you can because you need to move on to the next one. Because of that, we're typically bias ourselves towards visually scored traits. Lower cost in the sense that the upfront buying instrument can be hefty, but once you have it, typically it can run for many years. And I don't want to knock the undergraduates, but machines don't get hung over, they don't complain about the heat. So it reduces that layer of complexity in the data as well. And so the work I'll be talking about today was the work that was done in Maricopa, Arizona, in conjunction between the University of Arizona as well as the USDA, and kind of their pioneering phenotyping work using this platform that you see in the photo. And so Maricopa, Arizona is really an ideal place to do this type of work. We have what can be considered extremely consistent environment. It's always clear skies, it's always hot, it rarely rains, and this makes for a really, beneficial place to do this type of research. And so in that graph on the right, you can see that red line is 32 degrees C. Above 32 degrees C is when cotton really suffers a loss of efficiency in terms of rubisco-fixing carbon. And so in these 3 years that this trial is run, there's only 10 days where the temperature does not go above 32. And so because of that, we have this nice consistent environment. The other key component is this managed stress. There's a precision irrigation system, there's a few of them, allowing us to really finely control how much water the plant's receiving. And for this work, the water limited treatment as well as well watered. The water limited basically received 50% of the daily evapotranspiration rate of the well watered. And this was applied using subsurface drip irrigation. And just to note that the irrigation treatment itself was not started until 50% of the plots had already begun to flower to reduce the effects of, resources for real mapping. And it just so happened that TM1 became the reference genome that was recently sequenced. Just the basic field design, alpha lattice, the plots were, 9 meters long. So we had a good sample size. You can see how they're arranged in the field in terms of staggering wet, dry. And the phenotypes that we're looking at for the canopy are temperature, NDVI, and height, which I'll describe shortly. Physiological trait data, abscisic acid content, and carbon isotope discrimination, 2 hallmark traits for drought phenotyping. And then, additionally, we looked at agronomic traits like lint yield, boll size, as well as fiber quality, and then some seed ionomics data, which I'll talk about in the second part of the presentation. And so here's the actual platform that was used. It's not nearly as sexy as some of the things that have been designed recently, but this was developed in 2010, and kind of just a basic set of sensors. Active multispectral canopy sensor, the crop circle, ACS 470, which some of you may be familiar with, which was measuring canopy reflectance. Infrared thermometer, as well as a sonar to get canopy height. We had onboard data loggers to collect everything, provide the georeference and timestamp. And then finally, the one piece that really makes this all possible is this GPS RTK system. Standard GPS has a resolution of about 1 meter.
But you can see in this photo, 1 meter either way might put you in a different row, or i.e. a plot. And so with the RTK system, you actually achieve that sub-centimeter level precision, which allows us to tie each data point back to a specific point in the field, i.e. a plant. And so with this platform, it cruises along about 3 kilometers an hour. This translates into phenotyping about 450 plots per hour, which is pretty good. I don't care how many undergraduates you did hire. I think you'd have a hard time beating that efficiency. And so again, just kind of to illustrate this, it's what we're really trying to capture is a longitudinal assessment of phenotypes over time, how these traits are developing in the growing season. But it's also critical to remember that what we're measuring is the plant's response to the stress, not the stress itself. And because of that, that is why it's so critical to have a managed environment, i.e. that we know that these plants, the only thing that they were suffering from was a lack of water. It wasn't like a nutrient, it wasn't biotic stress. And why this is important is for traits like canopy temperature. Canopy temperature is just like when you go to the doctor and they take your temperature. They either tell you that you have the flu or you might die tomorrow. I mean, it's not really indicative of anything. And so that's why it's critical to know what's going on in the field, so you can understand, what that phenotypic information is telling you. And so in this case, for canopy temperature, it was the plant-water relations. We also used NDVI. Most of you are probably familiar with NDVI as a quantification of biomass. However, in this context, we're using it to actually measure the canopy architecture in the sense that, when plants are well watered, they have full turbidity, the leaves are actually the incoming light. But as they lose water content, you can see in this photo they begin to droop. Because of this, it's not reflecting as much light. What we're trying to do is actually capture that signal using NDVI. And then finally, we have canopy height and leaf area index, which kind of gives us an idea of the whole plant response to heat and drought. And you can see in the bottom that, again, the data collection was initiated at flowering. That's when the drought stress was imposed, and was continued pretty much up until harvest. So here's what the actual geoprocess canopy temperature data looks like. And so each circle represents one canopy temperature data point, and you can see that blue means it's cooler, green means it's a warmer temperature. And so the first thing you can see is obviously the differences between the well-watered and water-limited treatments. But also you begin to see some of the fine-scale differences between the genotypes themselves, as well as the field environment, though. You'll notice that down here in this part of the field, even though this was a water-limited treatment, it's quite blue, and that's because that part of the field had a high clay content, i.e. water holding capacity. So those plants weren't as stressed as we initially thought they were. But again, overall, this gives us a very fine-scale view of what's occurring in the field as well as the genotypic differences. So as all the graduate students know, that to breed, you have to have variation for that trait, and it must be heritable. And so that is one of the first tenets and basic questions we had to answer. And for canopy temperature, we saw, very nice distribution. I do want to point out that the temperatures reported, these are relative, they're not absolute. If the plants are at 45 to 50 degrees centigrade, they'd practically be on fire. So just as a note. But you also notice that the heritabilities were quite high, 0.83, 0.9. So this gave us a good idea that we had, fairly good experimental control or collecting good data. But what we're really interested in, and why phenotyping itself is cool, is that we could collect this data at multiple time points throughout the day, 7, 10 a.m., 1 p.m., and 3. And so if we start at canopy height, you pretty much see the phenotype that you would expect, that plants don't get taller or shorter across the course of the day in response to drought. As we move up to the more responsive traits like leaf area index, you'll see there's a slight decrease over the course of the day if you have like a really good eagle eye. But if you look at NDVI, you'll see from 7 to 10, things are doing okay, but it's that 10 a.m. to 1 p.m., that's when the plant, begins to wilt. And that's that change in architecture I was talking about when the leaves start to droop. So we're actually able to quantify that. And then also, if you look at the well-watered, you'll see that actually NDVI slowly increases over the day, and this is from the actual diheliotropism, when the leaves are actually tracking the movement of the sun across the sky. And then finally, you look at canopy temperature. This has been a hallmark graph kind of produced numerous times before. At 7 a.m., things are pretty much isothermal, but by 1 p.m., you can see that the water-limited treatment's 10 degrees warmer than the well-watered. Now, the critical thing to point out is it's not the point measurements themselves that we care about. That's not the phenotype. The phenotype of interest, and what's really intriguing, is that for the first time, we can get the change over time. That slope, basically. And what this represents is the plant's dynamic response to the environment. And this is why high throughput phenotyping is becoming, allowing us to answer a more complex question is we want the plant's response to the environment. That's what we're interested in. And so by having this technology, we could use this as a new phenotype for our QTL mapping. And so for the rest of the talk, just for sake of brevity, I'm just going to focus on canopy temperature. And so given that previous graph, the question was, well, maybe that was just like a standout day, that we got really good data. It was extremely hot. Vapor pressure deficit was very high. But sure enough, when we looked at all the other time points in the season, irrespective of the stage that the plant was in terms of development, whether it was flowering, bull fill, or fiber development, we saw that same dynamic response of canopy temperature in the irrigation regimes. So that was very positive, that we're collecting good data that's hopefully capturing some biological signal. When we move on, we look at the canopy temperature readings themselves, and what became apparent was that they're more correlated within plant growth stages than across, because the idea would be that surely if there was no signal, it would just be linearly correlated across the season, i.e. due to proximity of the measurements. But what we saw was very distinct, groupings that pretty much mapped up with the developmental stages of the plant. This was exciting because it gave us, first indication that the platform was collecting some type of data that had real signal in it that was capturing the biology. It also really illustrates the point of why continued phenotyping throughout the season is needed. If you'd only picked one time point, you would have missed pretty much the entire story and not really capture what's going on in the field. And so using this was, kind of good validation and gave us hopes for the actual QTL mapping. Now I recognize this figure is a little overwhelming, but let me just walk you through it. So the two panels, on the top is the dry treatment, on the bottom is the wet, the well-watered. On the right-hand side are the 3 years in which data was collected. The left-hand axis are the individual days. And so again, those individual days, we're getting that slope across, over time as the phenotype. And then on the bottom are the actual individual chromosomes with the genetic markers. If you see a red smudge, I guess, those are alleles that are increasing the canopy temperature. If you see blue, that means it's decreasing it. And so the first thing we found was that we could detect specific QTL that had a very defined temporal expression. That these, were detected at the same time of the year in the 3 years. So this was super exciting. This was the first time we had empirical evidence supporting what we all thought in terms of, we know that genes just aren't turned on at the beginning of the plant's life and left on. That there is this coordinated expression, whether it's, due to developmental or whether it's in response to the environment, that there is, controlled expression of these genes. The other thing that's important to remember, though, is, we're in this age where sensors are cheap, generating data is easy. Everybody can do it. The question we have to keep in mind is the data that we're generating biologically relevant? Is there something supporting, what we're seeing? And so one of my concerns was that we're maybe just mapping noise, that we were duped by our own machine. And so those other traits I was telling you about in terms of ABA and carbon isotope discrimination, the QTL for those mapped to the exact same spots as those for canopy temperature. This was very encouraging to have, validated phenotypes basically serving to say that, yes, what this platform is capturing is relevant in terms of canopy temperature and the plant-water relations. There's that saying, I'd rather be, lucky than good. And 2 weeks after I finished this analysis, the cotton genome was published. And so when we took these QTL intervals and blasted them, sure enough for the ABA, we found an abscisic acid response binding element factor, which was excellent news. And then for carbon isotope discrimination, same thing, a drought response family protein. So again, this really serves as validation that there is biology underlying the data that we're collecting. I think as we move forward, in terms of all the sensors that are being developed and high throughput phenotyping technologies that come up, it's always important to kind of keep that context in mind. So finally, the one question was, well, if this data is capturing biology, can we use it to actually predict traits that we're interested in? And so lint yield. And so we used all this, canopy temperature data in 2012 to try to predict lint yield. And our predictions, I would argue, were fairly good for just using phenotypic data. There is no marker data used in these analyses. And so on the dry treatment, we reached a correlation of 0.7, which wasn't bad, but it raises the question again is, what's that actually mean? Like, what is driving that relationship? What is the underlying biology? And so we go back and begin to kind of tease this apart. The first thing we did was look at the 4 individual traits in terms of canopy temperature and DVI, and we used random forest regression to kind of pull out which factors were the most important ones driving this response. And so what we found was that, sure enough, canopy temperature was the overwhelming, predictor that was the best. Next, we look at the time in the season, those individual days, and you can see days 201 and 208 are vastly, more important than the other, and this is when flowering is occurring. And intuitively, this makes sense. I mean, you can't have a cotton boll form without a successful fertilization event. And we know that pollen is liable to heat stress. And so, again, this was just kind of important information for us to really, convince ourselves that what we're doing had some valid meaning. And then finally, when you look at the actual time points within a day, you see that 10 a.m. is just a little bit higher in terms of its predictive accuracy, or and why we think that is because at 10 a.m., you're starting to see good stress symptoms in the field, whereas the 1 and 3, it's so severe that the plants are just kind of blowing out. So at 10 a.m., we're kind of getting that more fine-tuned response. But the biggest thing is that canopy temperature at flowering peak bloom is the most predictive of lint yield. And this type of information from the producer standpoint can be used to kind of monitor and schedule how they're going to use their irrigation. Say if you only have a certain amount of acre feet that you can apply per year, so you'd probably want to use that during flowering. So now I'd kind of like to change gears and talk more about understanding environmental effects. We know that the phenotype is relying on the genotype, the environment, and their interaction. But pretty much since the dawn of man and plant breeding, we've done a really good job of phenotyping half the plant. And obviously that's the top half. In-field root phenotyping is in its infancy. I don't know how many people went to Phenome last week. There's a few technologies that are developing, but nothing at field scale that you could really employ. But that still doesn't answer the question and doesn't resolve anything. We have to figure out how to understand how the root system's interacting with the soil environment, because as that photo shows, and what we already know is that it's highly heterogeneous. There's a lot going on down there. And we, have, not by choice, but because of technical limitations, have kind of neglected that. And so one way we thought about approaching was this is we could have some type of proxy for the soil environment and include that in our analyses. And so for that, we turn to ionomics, which is the rapid profiling of elemental composition of samples. And in terms of biological samples, elements are an essential component of every cell. And specifically like for plants, chlorophyll, magnesium is hanging out in the middle of that antenna collecting all that light energy. And so because of that, changes in elemental concentration, due to abiotic stress like drought or soil composition can be quantified. And so this, ionomics data can actually provide us information on the overall health and function of the plant itself. And you can see, in this element, the periodic table, which elements we're phenotyping. The green boxes are the essential ones for growth. Purple will kind of trace. And so I guess just to highlight again that this type of information gives us more insight into the physiology of the plant than maybe we were, hoping for. And so in this work, we also performed ionomic analyses on the cotton seeds themselves, but we also did soil sampling throughout the field and analyzed that as well. And so just again, we know that the soil environment is highly heterogeneous. And so here is the profile of the field and where the real population was grown. And you can see there is quite a bit. In our case, we had the distinct advantage of using the same field for 3 years in a row. And so if you were to think about this in the context of multiple locations, different fields, the impact of the soil environment, that complexity would ultimately impact your phenotype. And so looking at magnesium, one of the key components of chlorophyll as well as other physiological processes, we can see that there is in fact this gradient across the field. We used kriging to actually develop this interpolated field map. And by doing that, we can go back and add plot level data for the magnesium concentration into our analyses. And so sure enough, when we did the analysis, we found that the magnesium concentration within the soil was highly correlated with what we saw in the seed. And so by being able to, A, know about that variation, B, control for it, we better serve to improve our genetic mapping as well as understand the biology in terms of how magnesium is used by the plant itself. And so going forward, once we've kind of controlled for the soil environment, was to look at the actual ionome itself. And these are the genotypic correlations. These are not the phenotype correlations. And what we saw is that we got this really strong clustering right in the middle of this network diagram. And that the biologically relevant elements were all grouped pretty closely together. They all had high correlation values. And then finally we had the micronutrients kind of on the perimeter. And you also see that potassium is hanging out there at the top. And we think the reason why potassium doesn't group with anything is because it's so prevalent in just about every process in the plant that there was no real, established relationship. But basically, this gave us our first indication that this ionome, that this biological system is highly, interrelated and integrated. And you can see that between the two irrigation treatments, there's no real differences, i.e. that the ionome responds basically as a unit. And so with that in mind, we had to think about how best to map this trait. And so, as basic geneticists and everybody, you take your traits and you analyze them one by one. And so we did that first. We used a Bayesian classification technique so we could fit all the markers at once to really control for the background effects because we were concerned about that given the correlation among the traits. And so you can see for the various elements, we've got multiple hits across irrigation regimes. But what we really thought was, well, if everything's so interrelated, we need to take that into account in our analyses for mapping. And so we turned to using a multivariate approach. And so the first thing we had to do was to decide how to group the elements themselves. And so based on the literature as well as some clustering, hierarchical clustering done using the genetic relationships, we broke things down into those elements that are involved in ionic reactions as well as redox. And so we use those as the group phenotypes and perform the mapping. And so the multivariate QTL mapping, detected the same, QTL that the individual elements map to. But more importantly, we were able to detect 9 more QTL that, at a significant threshold, that in the Bayesian analysis, although detected, weren't declared significant. So this shows us that by borrowing information through the correlated structure of multivariate analyses, we're able to improve our power to identify loci controlling this trait, as well as pleiotropic QTL, which in the context of the ionome makes better sense given how everything is so related. But again, it was kind of the same question that we had with, the canopy temperature data was, if this data is capturing physiology, surely it should have some predictive value, that we don't always have to rely on marker information. And so we took the 3 individual years and generated blues for those. We did the PCA and colored things accordingly to well water and water limited. And if you look at a 3D plot of this, it actually gets a very nice separation. So based on that, we thought, well, surely, if we use some supervised machine learning techniques that we could probably predict, the stress level, i.e. if it was water, well watered or water limited. And so using a host of like k-nearest neighbors, linear discriminant analysis, logistic regression, and support vector machines, we were actually able to, do cross-year prediction, use 2010 and 11 to predict 2012. And you'll see that the accuracy is surprisingly high, 97%. And I realize that this is a 2 classification problem, which is one of the simplest ones. But again, relying on, data from individual years in a field environment with no marker data, I would argue that this prediction accuracy was quite higher than I thought we would achieve. And so again, this just kind of harkens back to, I guess the theme is that it's important to understand what the data is telling us, what the biology is, that just collecting data in and of itself is not really, advancing science so much. We have to think about this in terms of our hypotheses and questions that we're asking. And so just kind of summarize with the field-based experiment, we had this novel platform that was able to measure dynamic traits under actual field conditions. Gave us this kind of first insight into temporal QTL expression, and provided us some indication of yield prediction. With the ionomics work, it was this ability to integrate environmental effects into our analyses and better understand how the environment regulates and drives the phenotype itself, and as well as the shared genetic basis and the ability to predict stress level. And so now I'd kind of like to spend, I think, the last 10 minutes of my presentation just kind of describing some of the future directions, not only my research, but kind of as I think the field is going. And so one thing that's been hampering us is that we've focused on traits at the macro scale level because, that's what technology really enabled us to do, in terms of measuring plant height, yield, flowering time. But really, the intrinsic traits that give rise to those occur at a much finer scale. And so the question is, how do we get at those? And so there's this idea of implantive phenotyping. And so we've developed this nanoparticle sensor that's actually taken up by the leaf through the stomata and resides in the intracellular air spaces. And it fluoresces based on the vapor pressure deficit inside the leaf. And this is with a group out of Cornell, Abraham Struck. And you can see here's a corn leaf. The circled area is where this, fluid with the nanoparticles has been injected into the actual maize leaf. So it sits there for a few minutes to equilibrate. And then when we look at it with the microscope, we get a very nice fluorescence pattern. And depending on the intensity of that fluorescence, we can actually quantify what the inside, what the relative water content is of that leaf. And so it's getting at these, very fine scale traits, I think is one of the areas that people are rapidly progressing. I know there's a group in Iowa State that has these wearable plant sensors, and they're kind of pushing the exact same thing, is that to understand the large scale traits, we need to look at the micro scale traits. The other one I would argue is whole plant imaging. I think this is already being done here at Texas, but one of the tenets of biology is that structure equals function. Nature is very good at designing the things that it needs to be successful. In this case, structure equals function equals a phenotype. And you can see this, I think this is actually a photo from the Texas Historical Society of this cotton plant. And you get this very distinct arrangement of cotton bolls on the plants. And recent work has shown that, the geometrical arrangement of plant parts on, the plant itself, it's not happenstance. That there is some kind of biological mechanism driving that. And so one of the questions is, can we use image analysis to really start to tease this apart? And specifically, the discovery of latent variables. That, as humans, the phenotype concept is very limited in terms of what we think of, what we define as a phenotype. And that actually may be a limiting factor because we haven't considered what other phenotypes might be prevalent that we're just not aware of. There was a group out of China that had an excellent piece of research that took images of plants, extracted over 400 phenotypes for drought stress, and the mapping results from that were far superior to just looking at standard traits like canopy temperature. So surely there is value. I mean, again, though, the other side of that is that as scientists, we want to know why. We want to know why we're seeing what we are. So although these black box approaches can be beneficial, it's always important to keep in context of what they mean. And so with this in mind, I was in New York at the time, and when you're in New York and the closest cotton plant is 2,500 miles away, and it's always winter in New York, you had to come up with basically a model. And so with a 6-pack of beer and some dowels and some cotton balls, I came up with this cotton plant with the idea of, can we image it and begin to do this type of work? And so I just used my iPhone, and I don't think I can play the video from up here. I think you guys have to. Okay, we can maybe just go to the next. Oh, perfect. And so anyway, this is just using an iPhone, very basic technology, stitching the images together. We get a fairly good representation of this model plant. And there was 28 cotton bolls on the plant, and all 20 are successfully, recreated in this 3D model. And what's exciting is that, we can begin to see the branching structure of the plant itself, or of my model plant, as well as the distribution of these bolls. And so That was all fine and dandy, but I mean, that's like best case scenario. That's, you know, sitting in your basement with an iPhone. And so the question was, can we do that with real plants? And so if you could play the next one. And so you'll see that you can actually identify closed bowls in this plant. There's, you know, open bowls as well. There's also cotton flowers that you'll see right here kind of towards the top. We get actual leaf architecture and arrangement. And this is what gets really exciting, is understanding how the plant itself is formed to solve these challenges of resource capture and development. You know, it's a very competitive world out there, and for plants too. And this idea that you need to be better than everybody else equally holds true. And so these different architecture and arrangements of structures become kind of critical in understanding the biology. And so the work I'm doing with like Ian Small and James Colhesi is kind of how do we scale this up to image entire cotton fields. And so that's an ongoing battle. Finally, I'll just make a quick pitch for envirotyping, that we're spending all this time phenotyping and genotyping to these exhaustive depths, but what do we do with our actual environmental data? You know, there's cheap technology right now to capture all this information, and I recognize that I haven't figured it out either in terms of how you integrate this into your actual analysis. I mean, it's overwhelming, right? I mean, you have not only the field site level, but you can go break it down the plot level. But that's the thing, is that we know that these micro environment factors impact the phenotype. I mean, our ionomics showed that. So surely when we look at, you know, position in the field in terms of relative humidity, these factors all combine to create the phenotype. And so, you know, I just basically making this pitch that I'm sure there's smarter minds than mine out there to actually begin to tease apart these questions and how we integrate this information. And kind of why this is important is it leads to this idea of developmental quantitative genetics. And it's that it's connecting gene expression to actual phenotypic expression over the course of the season. So plants reach a final phenotype, you know, by various methods. Not everybody has the same growth rate. Not everybody, you know, it's just like humans. We don't all progress at the same rate. And how we reach our final phenotype, there might be interesting patterns in that information itself, that development. But more importantly, it's figuring out how that developmental time series happens in the context of environmental variation like precipitation and temperature. And furthermore, tying in the genetic level of when those QTL are expressed relative to the plant development as well as the environment. And so this becomes the hallmark of precision breeding. You know, it's about the idea of having the right gene on at the right time in the right environment. And this is really what's capitalizing on in terms of, you know, G by E and how we maximize that. And so to really carry this work forward, it becomes the idea of data analysis to really connect all these different sources together as well as that environmental data. And so one way that I think a lot of people are thinking about doing this is integration through crop model or ecophysiological models. Is that we're really good at collecting environmental parameters. With high throughput phenotyping, we're now getting some of the traits that we're interested in terms of like leaf initiation rate, canopy temperature, chlorophyll fluorescence, and final phenotypes like yield and quality. And it's the parameters from the models themselves, these light use efficiency, these are the traits I think we're all really after. This is what defines yield. This is what gives rise to the plant itself. And so I think these are really where we're trying to push to, because we know that crop growth is not an additive process. But yet all of our statistical models are largely additive. And so how are we really going to break that apart if the tool we're using isn't designed to incorporate that complexity? And so some of the work, you know, is using this for improvement of genomic selection capabilities and predicting in new environments. And so I know I've talked a lot, but here's kind of like how all this information is integrated together, that we have the genotype, G by E, and environment at the top of the figure. You know, genotypic information, I would say, is a slam dunk at this point. We're really good at collecting this, storing it, analyzing it. Those tools are available. Our genomic selection models are fairly robust. GWAS has been really worked out. Envirotyping, you see on the other side of the figure, I would say we're largely pretty good at this as well. You know, it's just breaking this down to a smaller scale. And then we have the phenotype itself, and you'll see underneath there's I guess that bang symbol with field-based high-throughput phenotype. This is really what's going to allow us to connect the two together. And crop models really offer us a way to integrate all this information. And you can see that using the model parameters from the crop models as input into our genomic models, you can give us, that deeper insight in these actual biological traits that are driving the phenotypes that we observe in the field. And so, again, combining all this information, it's the ability, like in plant breeding, what's one of the limiting factors is the number of sites that you can evaluate your genotypes at. It's always a challenge. It's the most costly part. But what if you could model, if you had historical data and the current data, and you could get a fairly accurate model of what that phenotype was going to be like, that could rapidly speed up your genetic gain. And I'm not going to stand up here and say it's all about these modeled phenotypes, that through each process, these tools and information, the synthesis of this, can answer basic biological questions that we've had for quite a while. And so I just like to conclude that, in terms of increasing genetic gains, it's not new concepts. That it's just new tools that are available. And we have to kind of change our mind in terms of how we're going to incorporate that. Like for genomics, GS and GWAS, both of these depend on having high quality phenotypic data. And with phenomics, we're able to get that more and higher quality data. So when we think about how we combine these, whether it's through crop models or something to be, found yet, it's just this idea that to really synthesize biological insight from data, we need to focus on our analytical capabilities and this is going to be a challenge. It's not easy. We're in the age of data overload, and there are no, simple solutions anymore to these problems because the complexity is so hard. And so with that, I'd just like to conclude there's always a ton of people who think on projects like this, specifically the Gore Lab at Cornell University, the USDA in Maricopa, Donald Danforth Center for doing the actual ionomic analysis, helping with that. And as a final slide, if anybody's looking for a postdoc and would like to live in Tucson, Arizona, please let me know. And with that, I take any of your questions.
Thank you, Dr. Pauli, for your presentation. If anyone has questions, please raise your hand, and we will have a volunteer come up to you with a microphone. We have a question from the webinar.
This is actually from me. Regarding the vapor pressure fluorescence system, how does that work with regards to physiological levels that we expect to see? Is it all throughout the range, or is it just limited to a certain portion?
Well, the relative humidity within inside a leaf ranges between 99 and 100% humidity, right? It's a very defined range. Because if you don't have that, the gas exchange breaks down. So with that nanoparticle sensor, it is tuned for that, pretty much those conditions.
So it was just water level, or I guess I was thinking it was other volatiles as well?
No, it's just purely water.
Purely water. Thank you.
Any more questions?
Hey, Duke.
Oh, boy.
So you spent a lot of the beginning of your presentation presenting QTL that you've mapped, and then the end, you were really talking about how we're going to use various information, but you didn't really cover how you might use those QTL. Do you anticipate using those QTL in any type of way in the future?
Absolutely, right? Detecting a QTL is only but half the battle. It's figuring out what that, what is that signal, right? What's the gene underlying that? In terms of breeding, the QTL that we identified, those would be obvious candidates in terms of marker-assisted selection if we had the marker, we had confidence where that marker was in relative to QTL. But yes, in terms of the applied breeding, I would want to incorporate that information into, picking parents and progressing those traits in advanced germplasm. From the more basic science side, I think it goes back to, again, understanding what gives rise to those QTL, because the QTL in and of itself is not the story. I hope that answered your question, Seth.
We have another question here. Dr. McGill.
I was wondering if this uniformity of year to year where you are actually cuts down on the ability to detect QTLs and so forth that might have an effect otherwise.
Certainly, yes. Right, how much variation do you want to see in your environment? Yeah, there is that, I guess there would be that argument that because it was so uniform year to year, that maybe that hurt us. But at the same time, you have to keep in mind that our mapping population of only 95 lines, which admittedly is very, underpowered in terms of detecting QTL, it was kind of ideal. Because if we would have had too much variation with that small sample size, we might have been hosed and not seen anything. So I think in this context, yes, but I mean, going forward, obviously I'd always push for more environments in different locations.
Any more questions? Two hands in the back of the room.
Thank you. Sorry. So going back to your data that you were talking about, and you've obviously collected gross amounts of data. And how are you, as you collaborate with across institutions and within, centers and such, how are you sharing your data, and how are you managing that data on a, obviously a lot of it?
This is going to be an embarrassing response, but when I started my postdoc, I was given 8 gigs of Excel spreadsheets. And unfortunately, that is where we were at the time. And I spent the first 6 months of my postdoc just doing that. And so you're absolutely right. I mean, when we think about phenotyping, it's the data question. Where is it going to live for the rest of its life? Who's going to curate it? Those are very valuable questions. So yeah, I can tell you with ours, it's sitting on my computer. If somebody was interested in using it, I'd be more than willing to share it. But I know probably not the most in-depth.
If you have USDA funding, do you have to make that data associated with your research publicly available, or is it?
Yes, although most of this project was funded by the NSF, which still mandates the same. Yeah, and so the data itself is public in the publications that are out there. However, I mean, if you contact us, we have no problems providing. I would just, it's like what repository to use. I mean, Cyverse, or what used to be iPlant, was a great avenue for hosting data and still is. I imagine at some point that's where all the phenotyping data will migrate to.
Yeah. My question is that, do you have any idea or what causes for the temporary QTL you identified?
Right.
Do you have any idea on that?
What underlies those? There was in terms of the QTL interval itself, when we blasted that, we found it had a few candidate genes that looked okay, but nothing that was concrete, and certainly I wouldn't hang my hat on. So no, we haven't figured out what those are.
You think that's a variation in the gene? Because some gene may be in different stage, maybe have a different function.
Sure, right. And
also, certainly, there's an RNA alternative splicing issue. That means different transcript, the same gene. Maybe different transcript, maybe response, different temporary QTL as well. You think that makes sense?
I mean, absolutely. I mean, it even comes down to basic, transcription factors in terms of what's needed at the time. And so, yeah, again, I apologize. We didn't have the depth of data to answer those questions. But very interesting nonetheless, right? Because that's ultimately what we want.
Thank you.
Any more questions? Do we have any questions from the webinar? If not,
Perfect.
Let's thank
Thank
Dr.
you.
Pauli once again. Our next speaker today is Dr. Sindhuja Sankaran. Dr. Sankaran is an assistant professor in the Department